Well, everyone, I'm Ralph Clayton, host of the VTM Podcast. This is episode 18. Today, I want to take you into one of the most fascinating frontiers in modern medicine: regenerative medicine, tissue repair, stem cells, genes, and the ancient developmental instructions hidden inside the body. This is not simply a story about healing wounds. It is not simply a story about replacing damaged organs. And it is not only a story about science trying to make the impossible possible. At its deepest level, this episode asks one dangerous question. What if the body has not forgotten how to rebuild itself? Because every body is already a miracle of construction. Before we are born, a single cell becomes many. Those cells become tissues. Those tissues become organs. A heart begins to beat. Nerves find their paths. Bones take shape. Fingers separate. Skin closes. Eyes form. Blood vessels branch like rivers. And somehow, without conscious instruction, the body builds itself. This is the mystery at the heart of regenerative medicine. The question is not only whether medicine can repair the body from the outside. The deeper question is whether medicine can learn how the body once built itself from within. Because healing and regeneration are not the same thing. Healing closes damage. Regeneration replaces what was lost. Healing protects the wound. Regeneration restores the pattern. Healing says, stop the bleeding. Seal the breach. Survive. But regeneration says something more astonishing. Remember the form. Rebuild the missing structure. Restore function. Make again what was taken away. That difference matters. When human skin is cut, it can close. When the liver is damaged, it can restore much of its mass. When blood is lost, the marrow can produce more. When bone breaks, it can knit itself back together. The body is not helpless. It is full of repair systems. But when a human loses an arm, the arm does not return. When a spinal cord is severed, the lost connection does not simply rebuild itself. When heart muscle dies after a major heart attack, the body often replaces living tissue with scar. When cartilage wears away, it does not easily restore itself. And that is the boundary regenerative medicine is trying to cross. Not merely wound closure, not merely survival, but true restoration. The dream is ancient. Human beings have always imagined bodies that could return from injury whole. Mythology is full of renewal. Serpents shedding skin. Gods restored after dismemberment. Heroes healed by sacred waters. Creatures that lose a part and grow it back. But modern biology has made the dream more precise. Because there really are animals that regenerate with astonishing power. Salamanders can regrow limbs. Axolotls can restore complex structures after injury. Zebrafish can regrow fins and repair parts of the heart. Planarians can rebuild entire bodies from fragments. Even mammals, including mice and humans, retain a small echo of this power in limited fingertip regeneration under certain conditions. So the question becomes sharper. Why them? Why not us? Why does one animal rebuild a limb, while another seals the wound with scar? Why does one body remember the missing pattern, while another rushes to close the injury and move on? This is where regenerative medicine becomes more than medicine. It becomes developmental biology in reverse. Because to regrow a limb, the body must solve many problems at once. It must close the wound without shutting down. Growth too early. It must control inflammation without allowing chaos. It must summon the right cells. It must tell those cells what they are. It must tell them where they are. It must tell them what is missing. It must rebuild bone, muscle, tendon, skin, blood vessels, and nerves. It must restore scale. It must restore orientation. It must know front from back, top from bottom, left from right, tip from base, and then it must stop. That last part is crucial. Regeneration is not just growth. Cancer is growth. Scarring is repair. Swelling is response. Regeneration is ordered growth, patterned growth, growth that remembers the body's architecture. That is why this field is so difficult. It is not enough to make cells divide. The cells must divide in the right place, at the right time, into the right tissues, in the right shape, and then obey the signal to stop. The body must become, for a moment, an embryo again, but only in the wound, only where needed, only long enough. That is the central danger. To unlock regeneration, medicine may have to touch some of the same deep programs that built the body before birth. Those programs are powerful. But power without control is not healing. It is risk. This is why genes matter. Genes are not simple switches. They are not magic buttons. They are instructions, regulators, timing systems, and relationship networks. A gene can help tell a cell what proteins to make. A transcription factor can help control which genes become active. A signaling molecule can tell nearby cells how to behave. An enhancer can help determine where and when a gene is turned on. And a developmental pathway can coordinate whole populations of cells into tissue. Regeneration depends on these networks. Not one gene alone. Not one stem cell alone. Not one injection alone. A limb is not a single tissue. It is an organized society of tissues. Bone without muscle is not a limb. Muscle without nerves cannot move. Nerves without targets cannot restore sensation. Blood vessels without structure cannot sustain the new tissue. Skin without pattern only covers. Tendons without anchors cannot transfer force. Joints without alignment cannot serve motion. This is why limb regrowth is one of the hardest problems in medicine. To regrow a limb is not to grow flesh. It is to restore a living machine that is also part of a person. Touch, grip, balance, pain, temperature, identity. The hand is not only anatomy, it is expression. The foot is not only support, it is freedom. A limb is biology, but it is also life. And that is why the promise of regenerative medicine is so emotionally powerful. Modern prosthetics have become extraordinary. Bionic limbs can restore movement. Sensors can help with feedback. Brain-machine interfaces are advancing. Materials are improving. Robotics are becoming more refined. But even the most advanced prosthetic is still a replacement. Regenerative medicine asks whether someday the body itself might become the builder again. Not metal, not plastic, not external machinery, living tissue, living sensation, living repair. That future is not here yet, but pieces of it are becoming visible. One of those pieces is the blastoma. The blastoma is one of the key structures in limb regeneration. In animals that regenerate appendages, after amputation, cells gather near the wound and form a growth zone. This is not simply a lump of undifferentiated material. It is a regenerative command center. It contains cells that can proliferate and contribute to rebuilding missing tissues. In salamanders and axolotls, the blastoma is part of how the animal reconstructs the missing limb. It is the place where injury becomes pattern, where wound becomes possibility, where the body stops merely sealing the damage and begins rebuilding the absent form. This is one of the great differences between regeneration and scarring. In a strong scar response, the body acts quickly to protect itself. That is useful. Scarring prevents infection. It stops fluid loss. It creates closure. But the scar can also become a wall against regeneration. It says, the emergency is over. The boundary is sealed. No further reconstruction is required. For survival, that makes sense. For regrowth, it is a prison. So regenerative medicine must learn how to change the wound environment. Not ignore the wound, not prevent defense, but guide the wound away from permanent closure and toward controlled reconstruction. This is why inflammation matters. Inflammation is not simply bad. It is part of healing. Immune cells clear debris. They release signals. They help coordinate repair. But inflammation can also push tissue toward fibrosis, scar, and chronic damage. The immune system is not only a defense system. It is also a construction supervisor. If it gives the wrong orders, the wound becomes scar. If it gives the right orders, regeneration may become possible. This is one of the reasons stem cells alone are not enough. For years, stem cells became the public symbol of regenerative medicine. And there is a reason for that. Stem cells can produce specialized cells. Embryonic stem cells are pluripotent. Induced pluripotent stem cells, or iPSCs, can be made by reprogramming mature adult cells into a more embryonic-like state. Adult stem cells help maintain and repair specific tissues. These are powerful tools. They allow scientists to model disease, grow organoids, test drugs, study development, create specialized cells in the laboratory. And in some areas, cell and gene therapies are already real medicine. But the public imagination often ran ahead of the science. People heard stem cells and imagined a universal cure. A knee repaired overnight, a spine restored, a brain renewed, an organ replaced, a limb regrown. But a stem cell is not a magic seed. A cell needs context. A cell needs signals. A cell needs a niche. A cell needs blood supply. A cell needs mechanical forces. A cell needs immune compatibility. A cell needs instructions, telling it what to become and what not to become. Without control, cell growth can become dangerous. Cells can go to the wrong place, become the wrong type, fail to integrate, trigger immune responses, or form tumors. That is why real regenerative medicine is not just about adding cells. It is about directing systems. This is the fourth lesson. The future of regeneration will not be one technology. It will be a convergence. Genes, cells, scaffolds, bioelectricity, immunology, developmental biology, gene therapy, tissue engineering, organoids, biomaterials, mechanical loading, nerve guidance, vascularization, and precise control of timing. A new limb, or even a complex tissue repair, will require an orchestra, not a single instrument. This is where developmental biology becomes central. Developmental biology studies how bodies are built, how cells know what to become, how tissues form shapes, how patterns are established, how limbs bud from embryos, how fingers separate, how bones lengthen, how muscles align, how nerves wire into targets, and how the body produces structure from information. If regenerative medicine is the goal, developmental biology is the map. Because to regenerate a tissue, scientists must understand how that tissue forms in the first place. This is why limb progenitor cells matter. In development, limb progenitors are early cells that help create the limb. They carry a kind of biological commitment. Not yet bone, not yet muscle, not yet tendon, but already part of the limb building program. Recent research has identified combinations of transcription factors that can push certain mouse cells toward limb progenitor-like states. That matters because it suggests scientists are beginning to learn not just how to make generic cells, but how to give cells regional identity. Limbness, not just growth, not just stemness, but limb identity. That word matters. Identity. In regeneration, the body must know what is missing. A hand cannot grow where a foot belongs. A fingertip must know it is a fingertip. A bone must know its length and orientation. A nerve must know where to reconnect. This is one reason the axolotl is so important. The axolotl is not just an animal that heals well. It is an animal that remembers position. When it regrows a limb, the new structure must match the old body. It must grow the correct parts in the correct arrangement. The wound does not simply produce tissue. It produces anatomy. And that is far more profound. Recent work has begun to identify conserved, genetic programs that appear across different, regenerating animals. Scientists studying axolotls, zebrafish, and mice have focused on genes such as SP6 and SP8, which become active in the wound epidermis during appendage regeneration. This is important because the wound epidermis is not just a covering. It is an instructive layer. It speaks to the tissue beneath. It helps coordinate regeneration. When key regenerative signals in that layer are disrupted, regeneration can fail or become defective. And when scientists use targeted gene delivery approaches to restore downstream signals, regeneration can be partially improved in animal models. This does not mean human limb regrowth is around the corner. That must be said clearly. A mouse digit tip is not a human arm. A zebrafish fin is not a human leg. An axolotl limb is not a clinical therapy. But these findings matter because they suggest regeneration is not pure fantasy. It has molecular language, and some of that language may be conserved. The body may still contain fragments of an ancient repair grammar. The challenge is learning how to read it, and then learning how to speak it safely. There is another powerful clue from experiments in adult frogs. African clawed frogs do not normally regenerate full limbs as adults, but researchers used a wearable bioreactor and a brief drug treatment applied to the amputation site. The treatment lasted only 24 hours, yet it helped trigger a much longer regenerative process. Over many months, the frogs regrew more complex limb structures than untreated controls, including tissues associated with function. This does not mean the same approach will work in humans, but it shows something important. Sometimes regeneration may not require controlling every step from the outside. Sometimes the right intervention may open the door, and the body may carry the process forward. That is a profound idea. Medicine may not need to micromanage every cell. It may need to create the right beginning, the right wound environment, the right signals, the right temporary state. Then the body's own patterning systems may do part of the work. This is one of the deepest hopes in regenerative medicine. Not to build a limb piece, by piece like a machine, but to persuade the body to resume an interrupted developmental program, to make the wound sight, permissive, to awaken the pattern, to let living tissue organize itself. But again, this is where caution becomes essential. A body is not a factory. A human patient is not a laboratory animal. A treatment that works in a frog may fail in a mammal. A treatment that works in a mouse may fail in a person. A treatment that regrows a small structure may not scale to a large limb. A treatment that creates growth may create pain, deformity, immune reaction, or cancer. This is why the field must resist miracle language. Because desperate patients are vulnerable. People with spinal cord injuries, degenerative diseases, chronic pain, arthritis, vision loss, heart damage, or limb loss may hear the phrase regenerative medicine and hope for rescue. That hope is human, but it can be exploited. There are clinics that market unapproved stem cell treatments, exosome products, amniotic products, and other so-called regenerative therapies for conditions where they have not been proven safe and effective. This is not a small issue. It is one of the moral shadows around the field. Real regenerative medicine is one of the most serious scientific frontiers of our age. But fake regenerative medicine borrows its language. It uses words like healing, natural, cellular, rejuvenation, and repair. It sells hope without proof. It turns suffering into a market. That is why regulation matters. Clinical trials matter. Manufacturing standards matter. Long-term follow-up matters. Evidence matters. Because in regenerative medicine, the danger is not only doing nothing. The danger is doing something biologically powerful without understanding what it will do. A bad drug can harm a patient. A bad regenerative product can become part of the patient. That is a different level of responsibility. This is especially true when gene therapy enters the picture. Gene therapy can deliver instructions to cells. It can change what cells express. It can modify biological behavior at the site of injury. That makes it potentially powerful for regeneration. But it also raises questions. Which cells receive the signal? How long does the signal last? Can it be turned off? Does it spread beyond the target site? Does it create unwanted growth? Does it alter immune behavior? Does it affect cancer risk? Does it work the same way in older patients? Diabetic patients, injured patients, or patients with chronic inflammation? These questions are not reasons to abandon the field. They are reasons to advance carefully. Because the prize is enormous. Imagine scar, free wound healing after burns. Imagine cartilage restored before joint destruction. Imagine heart tissue repaired after injury. Imagine nerves guided across damaged regions. Imagine diabetic ulcers closing with healthy tissue instead of chronic wounds. Imagine craniofacial defects repaired with living bone and soft tissue. Imagine organoids grown from patient cells to test therapies before they are used. Imagine children born with tissue defects receiving repairs that grow with them. Imagine one day, far beyond today's capabilities, partial limb structures restored with living sensation. That is the horizon. But the path to that horizon is not straight. It passes through many smaller victories. Better wound healing, better skin regeneration, better nerve repair, better vascularization, better bone regeneration, better control of fibrosis, better cell delivery, better biomaterials, better immune modulation, better developmental maps, better safety switches. Before medicine regrows a human arm, it will likely master smaller forms of repair, a fingertip, a joint surface, a tendon, bone interface, a nerve bridge, a heart patch, a tooth structure, a retinal cell layer, a skin graft that restores glands and hair follicles, a cartilage defect that heals with real cartilage rather than scar, like substitute. These may sound less dramatic than full limb regrowth, but they are not small. For the patient who can walk without pain, cartilage matters. For the burn survivor, skin quality matters. For the person with a damaged cornea, vision matters. For the patient with a failing heart, living repair matters. Regeneration does not need to begin with miracles to change lives. It can begin with better tissue, healthier repair, less scarring, more function, more dignity. That is the realistic power of the field. And then, step by step, the harder questions can be approached. How do you regenerate complex shape? How do you restore nerve integration? How do you scale from small structures to large ones? How do you make new tissue vascularized fast enough to survive? How do you coordinate multiple tissue types? How do you make regenerated tissue stop growing? How do you prevent immune rejection? How do you make therapies affordable? How do you manufacture living treatments consistently? How do you regulate a therapy that is part drug, part device, part tissue, and part developmental instruction? These are not minor obstacles. They are the real road. This is why regenerative medicine is both thrilling and humbling. It shows us that biology is more capable than we once believed. But it also shows us that the body is not a machine with interchangeable parts. A machine can be repaired by replacing components. A body must be restored through living relationship. Cells listen to signals. Tissues respond to forces. Genes open and close. Immune cells shape outcomes. Nerves influence growth. Blood vessels decide survival. The extracellular matrix gives architecture. Time itself becomes a treatment variable. Too early, and the wound is not ready. Too late, and scar has taken command. Regeneration is not only about what is delivered. It is about when it is delivered, and where, and for how long. This is why the next era of regenerative medicine may depend on precision, not the old fantasy of one universal injection, but precise biological choreography, a wand dressing that releases signals in sequence, a scaffold that guides cells spatially, a gene therapy that activates only in the right tissue, a cell therapy that survives, integrates, and then matures, a bioelectric signal that changes the patterning state of a wound, an immune treatment that shifts macrophages away from fibrosis and toward rebuilding, a personalized organoid model that predicts how a patient's cells will respond, a safety switch that can shut down unwanted growth. This is the future if the field succeeds. Not magic, choreography, not miracle, instruction, not forcing the body, teaching the body to remember. So what is regenerative medicine truly about? It is about healing. But healing is only the surface. It is about whether the body can be guided back toward its own deeper intelligence. It is about whether injury must always end in scar. It is about whether lost tissue can become a site of renewed pattern. It is about whether stem cells can be disciplined into architecture. It is about whether genes can be used not merely to correct disease, but to reopen repair. It is about whether developmental biology can become clinical medicine. It is about whether the body's first language can be spoken again after trauma. And it is about whether hope can be protected from hype. Because that distinction matters. Hope is necessary. Hype is dangerous. Hope funds research. Hope sustains patients. Hope asks difficult questions. Hope refuses to accept that today's limits are eternal. But hype sells certainty before evidence. Hype turns early animal studies into promised cures. Hype erases risk. Hype preys on grief. The future of regenerative medicine must be built on hope, not hype. On evidence, not slogans. On patients, not markets of desperation. On biology, not fantasy. And if it succeeds, it may change medicine at its roots. Today, much of medicine manages damage. It slows decline. It reduces symptoms. It replaces function externally. It cuts out disease. It suppresses inflammation. It stabilizes. These are necessary. They save lives. But regenerative medicine imagines another layer. Repair the tissue. Restore the function. Rebuild the structure. Return the body closer to wholeness. That is a different kind of medicine. A medicine that does not only fight disease. A medicine that studies creation itself. Not creation in the abstract, but the creation of skin, bone, nerve, blood, muscle, cartilage, tooth, heart, limb, the creation of form. The creation of function. The creation of the living pattern. And perhaps that is why limb regrowth captures the imagination so strongly. Because the limb is visible. Its absence is visible. Its return would be undeniable. A regenerated limb would not be a hidden biomarker. It would be a public sign that medicine had crossed an ancient boundary. But even before that day, the field may transform quieter forms of suffering, chronic wounds, degenerated joints, damaged nerves, burn scars, bone defects, dental and craniofacial injuries, retinal disease, muscle loss. These are the places where the future may arrive first, not as science fiction, as tissue that heals better than it used to, as scars that form less aggressively, as cells that integrate more faithfully, as organs modeled before they fail, as therapies designed from the body's own developmental logic. That is the heart of regenerative medicine. The body is not only material, it is memory. Not memory like thought, but biological memory, pattern memory, position memory, developmental memory, the memory of how to become. In ordinary healing, that memory is often limited. Close the wound, protect the organism, move on. But in regeneration, the body remembers more. It remembers what should be there. And the great scientific question is whether human medicine can learn to awaken that memory safely. Not everywhere, not chaotically, not forever, but precisely wound. At the repair. For restoration. For life. I'm Ralph Clayton, host of the VTM Podcast. Thank you for joining me for episode 18, and this look into regenerative medicine, limb regrowth, stem cells, genes, and the developmental biology of repair. And remember, not every healing is regeneration. Not every promise of renewal is medicine. And sometimes, the future of repair begins by listening to the body's oldest instructions.